we know about disease, particularly enteric diseases, involve complex interactions between the environment, the pathogen, and the host. So we've talked this morning a little bit about how environment influences pathogens, and then how pathogens will cause disease in the host. And there's been a lot of work, particularly on pathogen mechanisms and its influence on diarrheal disease. So most of the important enteric pathogens in pigs, we do know a lot about the mechanisms. But at the same time, we know that the presence of a pathogen alone is often not enough to cause disease. These are pathogens that are always in the environment. And that's because animals have these barriers that are in place, these biologic or physiologic barriers that protect them against everyday sort of bacterial or pathogenic challenges. Now, one area that is lesser known or less studied is how these animals become susceptible to pathogens. And a large part of this is the influence of the environment. And I'm going to talk a lot about stressors, and what we're studying in the lab, and how they can influence the host and make them more susceptible to pathogens. So again, two focus points in this talk. I am going to cover some of the pathogen interactions with the host that we've talked about already today, some of the mechanisms on how pathogens can take hold of the normal physiology in the gut in the pig and cause disease. But most of this talk is going to be focused on factors by which stressors predispose the pigs to become more susceptible to these pathogens. And I'll talk about some of the more recent data from my laboratory, which is a focus of my research, looking at how stress impacts epithelial barriers, how it negatively impacts immune function in the animal, and how when the stressors are occurring at certain critical periods, such as early in development, there can be some long-term or long-lasting effects on the susceptibility of the pigs. And at the end, I'm going to go a little bit into some of the mechanisms, biological mechanisms of how this may be working. So the intestinal tract, like I said, we're going to cover normal physiology first before we can understand pathophysiology. Here's a small intestine here. This is the colon. You can see that the intestine is lined by this single layer of epithelial cells. In the small intestine lining the villus, and then in the surface epithelium in the colon. And this epithelium serves critical but divergent roles in gastrointestinal health and function. On one hand, this epithelium must absorb a significant amount of nutrients, a lot of transport of water bidirectionally on a daily basis. But at the same time, it must provide a barrier because there are trillions of bacteria, antigens, and toxins residing in this lumen. The intestinal mucosa is the largest interface between the outside world and the body. So this function of this epithelial barrier is really critical because you have, again, all these toxins and bacteria and pathogens in this lumen literally micrometers away from the largest immune organ in the body, which is in the GI tract. So this is why we're so focused on some of these aspects of function of the gut, because once these are broken down, that's when disease results. So just kind of a review, I'm going to go over first just some mechanisms of diarrheal disease. If you look at the intestinal villus, the crypt-villus axis here, we know that there's functional differences in the activities of the gut. In the immature crypt epithelial cells and the stem cell populations, as these new epithelial cells are born, they migrate up to the villus tip and they eventually become mature absorptive enterocytes. We know that the immature crypt epithelial cells are predominantly secretory in nature. They provide and drive most of the fluid secretion in the gut, whereas the mature epithelial cells are more absorptive. But both epithelium can do both. Now, the relevance to enteric disease comes when pathogens can target certain areas of this villus here, say the mature epithelium here, resulting in loss of this absorptive surface epithelium, a characteristic of some damaging viruses or coccidiosis. Whereas, on the other hand, there are some agents and their toxins that can selectively cause disease in the ileum, in the crypt epithelium, eliciting a lot of chloride or secretory mechanism. I'll go a little bit more into the mechanisms, but this is what's driving a lot of the secretory diarrhea and fluid loss. So if you look up at this epithelium close up, this is lining the villus here. The gut is equipped with a lot of defense barriers in place to protect it from the external environment. There's secretory channels here that are secreting chloride and bicarbonate, which are for buffering, and fluid secretion, which are important for normal health. There's mucus being secreted. There's certain cells that are secreting antimicrobial peptides. We're learning more about this normal establishment of the microbiota and how that's really important. That's a barrier. There are also intrinsic components of this epithelium, including tight junctions. These are just clusters of proteins in between the epithelial cells lining the gut. And these are important because they regulate how permeable or leaky the gut becomes. And when the animal is under stress, or whether it's infectious or psychological stress, some of these proteins become degraded, and this starts resulting in a leaky gut, which is a susceptibility factor. Also, as a defense mechanism, there's immunologic barriers. We talked a little bit about secretory immunoglobulins and a resident immune system in the gut. So if these barriers become broken down, you need a rapid, robust response from the immune system to prevent the spread of this potentially systemic infection. And this is all regulated in large part by the enteric nervous system, in turn controlled by the circulation. So these are all very important defense barriers. So we're going to talk first about secretory and absorptive functions of the gut and how they become manipulated by certain pathogens. So if we look at the secretory mechanisms, and not to get too much in the detail here, is just know that one of the major mechanisms of secretory mechanisms in the gut is chloride secretion, and it involves certain channels, certain signaling pathways in the gut. And what this does, this drives chloride and creates this osmotic gradient with sodium, and then water follows. And this becomes important in certain enteric pathogens, such as Enterotoxigenic E. coli, which has toxins that stimulate these secretory pathways. Some viral toxins, other than damaging the epithelium, they can also, they've been shown to modulate other chloride channels in the gut that can induce some secretory mechanisms. And then there's other downstream effects, whether it's bacterial toxins or inflammatory products from a disease situation, which are going to elicit a lot of this secretory mechanism in the gut, resulting in a lot of fluid loss. So just an example, as I mentioned before, Enterotoxigenic E. coli. It is an organism that produces enterotoxins, the heat-stable, heat-labile toxins. They bind to the epithelium, and they induce these chloride channels to open through signaling pathways. So that's what's driving the fluid secretion and resulting in diarrheal disease. And here's a picture just from one of our challenge models. You can see dilated fluid loops, some water kind of accumulating in the cecum here, just a very secretory diarrheal disease. And in the acute stages, there's very minimal sort of destruction of the villus. So the whole architecture is not all that disturbed in the early stages. On the other side, the mechanism of absorption here, this is all driven by sodium transport across the gut. So there's certain exchangers that pull sodium in, and this again drives a osmotic or hydrostatic gradient in the other direction, and then water follows that gradient as well. And there's other mechanisms, whether it's sodium-driven transporters, sodium nutrient coupled transporters, amino acid transporters. They're all driven by sodium being driven across that epithelium. And this has been therapeutically manipulated with oral rehydration solutions. So these are known solutions that have been saving lives of kids with rotavirus and enteric disease in third world countries. These are very effective. And they utilize this sodium gradient here by activating these channels in the ORS solutions, which contain salts, mainly sodium, and a sugar source. They drive these sodium transporters to draw sodium and glucose across this epithelial cell, and that's what's causing water to follow. So these are important tools that have been used to combat diarrheal disease. And there are certain pathogens that attack these certain absorptive mechanisms. Again, Enterotoxigenic, which is always talked about in terms of a secretory type diarrheal disease, through its same pathways can inhibit a lot of sodium absorption in the gut as well. And there's been a lot of work on rotavirus and its pathogenic mechanisms, also can affect some of these nutrient transporters as well. So this is an example of where the malabsorptive, where there's significant damage to that surface epithelium here, such as would occur in viral disease. We talked about PED, obviously TG rotavirus. They cause damage to this surface epithelium, and a lot of these cells are lost, and a defense mechanism of the gut is to contract, to reduce this surface area, and you're left with a very blunted surface area here. And this surface area is not very absorptive. It's got mostly immature enterocytes. It's kind of secretory in nature, and resulting in malabsorptive, predominantly diarrheal disease. And then I just wanted to cover one more type of mechanism of diarrheal disease, which is inflammatory diarrheal disease. And the best example of this is Salmonella Typhimurium. This is where this organism, again, binds to the gut, elicits all these pro-inflammatory signals, recruiting a bunch of neutrophils into the GI tract. And they cause a lot of damage, result in lesions. Some secretory mechanisms are in place there as well. And then they progress to a malabsorptive gut as these lesions become chronic. And this is what it looks like. You have normal villi in a healthy animal here, and then it basically looks like a colon after a couple days of Salmonella Typhimurium. You see all these inflammatory cells in here, reducing absorption. There's some secretory mechanisms, and that's resulting in diarrheal disease. So I covered this part. Again, it was just a quick review. Just looking at it from a pathogen's perspective and how it causes disease in the host. But now we're going to look at what are some of these susceptibility factors that are going to allow this animal to become susceptible to the pathogen, and what's taking place at the gut level. And when I say stress, I'm going to show you a couple models, but we know stress can be anything that disturbs the homeostasis of an animal. This could be mixing stress, heat stress. I'm going to cover a lot of my work on early weaning stress. And it could be infectious stress. And what we're finding out is that the response to all these different stressors from a physio or biological level is actually quite similar. There's a lot of same pathways being upregulated. So I'm going to be talking about weaning stress, but really, I think we can translate that to a lot of other stressors that pigs are under. So we've known for a long time that stress is this important environmental factor that predisposes all animals and humans to disease. There are hot areas of research in human medicine because a lot of the stress and anxiety that people undergo are linked with the onset of a lot of GI disease in people. So it's an opportunity to study diseases that have dual benefit of understanding of both animals and humans. So we can see stressors, they can result in range of clinical conditions, whether it's just poor weight gain, feed conversion, increased susceptibility to GI infections, diarrhea. It can have a significant effect on the immune response, whether it's hyperactivated or suppressed, and other inflammatory diseases of the gut. But even though that we've known that stress is playing such a major role in disease, our understanding of the biology of stress and disease susceptibility is really poorly understood. So this is the focus of my research laboratory at NC State. We're really interested in how stressors impact GI function and how that leads to GI disease susceptibility. And we utilize a number of techniques, all the way from live animal models, systems, to in vitro techniques to begin to unravel how stress is causing disease in the gut. So when I became very interested in stress in disease, I wanted to begin to study this in a model that is in the pig that is associated with a lot of stress. So weaning was definitely one of those periods where we know piglets undergo a lot of stress, because on one day they're all happy with their brothers and sisters, with mom, and then on the next day they're being moved. There's a significant amount of stressors. Even though those environments are clean, the animals are well taken care of, there's just an inherent amount of stress by these abrupt changes. And these are a few of them, whether maternal separation, there's changes in environment, transport stress, diet transition. These are all stressors that are put on a very young animal at a critical stage in its life. And as a result, the post-weaning period, there's a lot of health challenges, as you all know that. So we wanted to really begin to understand how do these production stressors cause or impact gut defense mechanisms that we talked about and increase disease susceptibility. So again, we covered this, the epithelium, the epithelial barrier, and all these components. And this was a focus that I wanted to really begin to study, because we know that when this barrier becomes broken down under a variety of different stress, the gut becomes leaky. And then you have unimpeded access now of pathogens that can get across this gut, and toxins that in turn activate the immune system, can cause a lot of acute inflammation. And what this in turn will do, it'll impair the ability of the gut to absorb nutrients and do its normal functions. And it'll also increase its ability or sensitivity to secretory responses, contributing to diarrhea. And if these stressors become chronic, we know that this immune system can also become overwhelmed as it's persistently activated from this leaky gut, and now you get immunosuppression, which is another factor that's going to impair the animal's ability to respond to an infectious challenge. So how do we study that? And this is where I admit it gets a little bit of like a science fiction movie here in our lab. But how do we study the permeability of the gut? And we utilize a technique called the Ussing chamber, where we take intestinal tissue from recently euthanized pigs or any animal, and we can mount them on these chambers here. This is an intestinal mucosa mounted on this chamber. And in this chamber, the tissue is bathed in a physiologic Ringer solution. It's oxygenated, it has nutrients, and the gut is viable. It remains alive. It can absorb nutrients, it can secrete, it can respond to an infection or a pathogen. It can do a lot of things that normally it's just basically outside the body. We're just supplying it with what it lost when we took it out, which is the blood supply. So this provides us with a very unique opportunity to sensitively measure these gut barrier functions in the gut. And we do that by looking at permeability probes, where we'll have the intestine here, and this will be the luminal side, and this would be where the villi are. And then we would put a radio-labeled or a labeled substance, a permeability probe, on one side of the tissue, and then measure over time how much of that substance starts leaking out across the tissue. So if there's an increased flux, it means that you have a leaky gut or a compromised barrier. And if there's resistant to transport of these paracellular markers, it means that you have an intact or healthy barrier function. So this is important to understand, because when I've shown you some of the data slides, we're measuring it in flux. So a higher flux rate means leaky gut, bad, more permeability, and a lower permeability is good. Another measure, we can do a number of other measures in the gut looking at absorptive function, and one of the particular measure is this resistance. It's really just some physics here with Ohm's law. The tissue is hooked up to electrodes, and we can measure ion transport events occurring across this tissue. And this really gives us some unique insight into the physiology of the gut in a very sensitive manner. So we first looked at weaning stress, where we took pigs, we weaned them at 18 days of age, 24 hours later, we euthanized them, put their intestine on these Ussing chambers to see how permeable they were, if this was an important factor here. So if you look at resistance, this is resistance to the flow of ions. You see a significant reduction in resistance. So what that means, that's like an inverse relationship to permeability. So that means that there's more leaky ion transport across the gut. But focus on this one. This is the permeability markers here. 24 hours after weaning, you get this significant elevation in intestinal permeability. So this stress has broken down barrier function in the gut. And this response is quite rapid. It can occur within 30 minutes where you start seeing some permeability changes, even before any type of inflammation starts. So this is a very acute, very sensitive response in the gut. So this is our first indication that stressors were really doing had a significant deleterious impact on one of the primary critical defense functions in the gut, so increased permeability. We had since gone on looking at different models of stressors over the years, looking at mixing and crowding stress, chronic heat stress, and we realized that this was a pathophysiologic marker of stress on the gut. So you see elevated intestinal permeability when animals were just put mixing and crowding conditions for 7 days. This was after 10 days of heat stress. You see this major elevation in the permeability of the GI tract. And then you start thinking about these stressors and what I've showed you about the importance of that barrier and preventing pathogens from getting across, and how that's also linked to nutrient absorption in the gut. And we've also done some studies as well looking at nutrient absorption, and this is where we put glucose or other nutrients on the luminal side, and we can measure their electrical ability to increase the current, which is directly related to nutrient absorptive function. So just look at these graphs as nutrient absorptive function, and looking at a control compared to a mixing and crowder stress or heat stress, you can see that not only the barrier function is disrupted, but now the ability of the gut is significantly impaired to absorb nutrients. So we then went on to look at some factors that may influence the stress response in this young pig. And we looked at weaning age first, because that was, we felt like it was a significant factor that could be determining how much stress a pig is under at that time. If you just focus on this graph here, this is permeability. We looked at pigs that were weaned at 16 days of age versus 28 days of age. And we looked at their response following weaning. So this is unweaned, early weaned pig. You see 24 hours after weaning, this significant elevation in intestinal permeability. But this response was significantly attenuated in a late weaned control. And it's not because they're responding necessarily systemically differently to a stress. We looked at cortisol levels. We looked at CRF concentrations in their blood. And actually, the late weaned pigs actually had higher cortisol and higher CRF. So they were actually had a more robust central stress response to it. But at the gut level, they were more protected. And then we also did looking at incremental effect of weaning ages and post-weaning barrier function. And as you can see, this is permeability of the gut. As weaning age is delayed, their post-weaning gut function is markedly improved as far as barrier function. Then we became really interested in, okay, we see these stressors in the gut that are causing permeability early in life. What's the long-term impact of them? Do they eventually recover from this stress and go on to be normal pigs? Or are there lasting changes in this barrier function? So again, we looked at late weaned animals, 28 days of age, or 16 days of age weaned pigs, weaned them, and then looked at different time points post-weaning, all the way out to 112 days, far beyond the weaning event, to see if their permeability changes recovered or did they persist. And we found out, this was actually a recent study we carried out as longer than we had been before, you get this significant increase in permeability, but even far out, way past the weaning event, the animals that had undergone early weaning have this significantly or disturbance in their barrier function. They have leaky gut that persists throughout basically their production lifespan. So it's something that's happening here that's an underlying factor. And I'll tell you that we look at growth responses in a lot, and maybe a lot of times we're doing it in clean conditions, but we never see growth differences in these guys. So it's not, growth rate is not a marker necessarily of gut health in these animals. They have this underlying defect, and I'm going to show you what happens when we put these pigs under different stressors later in life. So the first study we wanted to look at, okay, so looking at these changes, long-term changes in barrier function in the gut and its influence of early weaning stress, do they have any subsequent effect when these animals are exposed to normal routine production stressors later in life? Do they respond differently? So we looked at mixing stress at 54 days post-weaning, and then after 3 hours of mixing and mingling these late weaned pigs, early weaned pigs in different pens to stress them out, we looked at barrier function or permeability. And we saw quite a difference in the response between early life stressed animals versus late weaned pigs. You can see after stress, you get a significant increase in gut permeability in the early wean group, whereas the late wean group, they didn't respond to this. And this was sort of a mild stressor. They weren't all that stressed out. It was kind of a low-grade stress. But you can see sensitivity and some blinded fecal scores too. The gut in the early weaned animals have this persistent or sensitivity, hypersensitivity to a later life stress. We also want to look at whether these animals respond differently to an infectious challenge. So we did an E. coli trial where pigs were weaned at early weaned or late weaned. In this case, the late weaned pigs were about 24 days of age, weaning 23 to 24. Early weans were about 18 days of age. And we looked at how, this was about 35 to 40 days post-weaning, how they responded to an E. coli challenge. And what you can see here, the late wean pigs, yes, they responded to an E. coli challenge. They got diarrhea in response to the challenge. But if you look at the pigs that were early wean, they had a more severe clinical disease in response to a challenge. And this was 4 days post-challenge here, where we began to look at some things that were going on at the gut level. And if you look at gut injury, again, this is permeability, our marker here. The early wean pigs had significantly more severe gut injury in response to the E. coli challenge compared to the late wean pigs. We also wanted to look at the immune response in the gut to see if that was dictating what was going on from the injury or diarrheal clinical disease level. And we found something interesting. If you look at the late wean pig controls, they responded with a significant immune response. They had more neutrophils being recruited into their gut. They had higher levels of inflammatory cytokines, IL-6 and IL-8. Whereas the early wean pigs, this response was markedly suppressed. And it kind of threw me back a little bit because I was thinking that I would be seeing more inflammatory cytokines in the early wean pigs. But if you think of a healthy response to an infectious challenge, it is to induce an immune response. You need an appropriate immune response to protect yourself against disease and prevent it from spreading. So the late wean pigs actually recruited more neutrophils, which are important for clearing E. coli from the gut. And all these cytokines that are responsible for modulating immune responses that are critical for clearance of the pathogen and getting over and preventing that pathogen from spreading. So this would suggest that the early wean pigs are more susceptible, maybe in part due to they're immunosuppressed by the time they respond to a subsequent stress. This is just a histology figure. This is early wean control. You'd see all these E. coli adhering to the gut, very few inflammatory cells, whereas in the later wean pig here, all these neutrophils start showing up, and that's a good response. You need that response. It'll cause some damage, but at the same time, it's critical for clearing out the pathogen. So at this point, we've shown that early life stressors, and we've shown in other model systems where it's not necessarily just weaning stress, other types of early life gut injury lead to long-term changes in intestinal barrier function, so the persistent changes in permeability, heightened stress responsiveness. Animals respond differently to normal production stressors. And they also have different disease responses, exacerbated clinical disease to subsequent challenges, which may in part be related to an inability to mount effective immune responses to a challenge. So at this point, we wanted to learn a little bit more about what could be going on at the gut level that's in addition to this permeability. What was driving this? What were driving the changes when an animal is challenged early in life with an injury in the gut? So we first looked at just histology, and we really didn't see many changes in the histological makeup of the GI tract. The villi were normal. They weren't different from early weaned and late weaned. Inflammatory cells were not really different. Cytokines, pro-inflammatory cytokines were not really different in the gut. It just looked normal. But I became really interested in one particular cell type, which is the mast cell. And we all know mast cells. I'll cover a little bit more about mast cells later, but it's more allergy-type cells. But I just had an interest in this because I was reading literature from human patients with irritable bowel syndrome, which, again, it's not an inflammatory disorder, but they have a lot of mast cells and leaky gut. And so we did a stain for mast cells in the gut. And you can see all these stained mast cells. This is the villi here. You may not see it, but in the mucosa of the gut compared to a late-weaned control. And you can look at these mast cells. They're always found near barriers here, and they're releasing their products. They have a lot of pro-inflammatory and proteases products in the cell, and they release, and they can cause changes in permeability. And this is just a marker of mast cell tryptase. It's a Western blot, protein blot, just showing that there's higher levels of this major mast cell marker in an early weaned gut. So again, just to review a little bit of mast cells. Mast cells are really interesting cells. It's one of an immune cell that my lab has become really interested in because they're just immune cells that are found at barriers. They're like sentinel cells, and they're packed with all these important products, whether they're pre-stored histamine, which we know is important for allergic responses, proteases, inflammatory products, as well as all these other products that can be synthesized by these cells. And they're found at almost every barrier in our body, whether it's the gut barrier, respiratory epithelium, bladder, skin, blood-brain barrier. These are important immune cells, which we think are the primary signals or receiving signals from the stress response. And they've been involved in a number of diseases shown to be playing a role. I just highlighted the ones in red that would be more probably interested to this group here, but just GI disease, whether it's infections in the gut, immune responses to bacteria, they're important for viral responses, vaccine responses, just general overall pathogenesis. So we wanted to begin to test if this mast cell, we found it in high levels of numbers, but we wanted to see if it was really involved with any of the changes. So we treated early weaned pigs. We had late weaned controls, early weaned pigs. We treated them with a sodium cromolyn, which is a mast cell stabilizer drug. And we administered this drug to them and to stabilize the mast cells. And over 24 hours, we euthanized the animals and looked to see if it changed the permeability defect in the early stressed animals. And you can see here permeability, early weaning stress have elevated permeability, but this is significantly reduced when we can block the activity of this mast cell. Again, showing us that the permeability or barrier defects is mast cell related. Now I'm just going to give a little more insight as we're delving a little bit more into the biology of this stress response, on how we think that mast cells are being activated in the gut, in pigs, and in other animals. And we focus on this system. We know that the corticotropin-releasing factor system, I think we all know that this is a critical stress response regulatory system. It's part of the fight or flight in the HPA axis. And we know that it's active on different receptors, and it's involved in a variety of different stress responses that is known. But we really knew nothing about, or the field knows nothing about the impact of this system on immune cells, really. It's all been studied in the nervous system. So we showed that in early weaned pigs, this system, through levels of CRF in the gut, is elevated. The receptors for this system are elevated in the gut. And when you block this system by administering the animals a CRF receptor blocker, we can significantly restore barrier function in these animals. And what was interesting, we just did finishing up a recent study on Salmonella infection in pigs, and this CRF system is also upregulated in a pathogen stress as well. So it's playing an important role in gut health, and it's not specific to a weaning stress. So just to end up quickly, we're beginning to link this, just to give you some future directions where we're heading on how the CRF system is activating mast cells. This is an EM micrograph of a mast cell. You can see all its granules in the gut. After weaning, becomes all activated, releasing its granules. When you treat with a CRF receptor blocker drug, we can stabilize these mast cells, and that's what's linked to restoring barrier function. And we've, just for sake of time, we're further identifying this link between CRF and mast cells, and because we think that in a stressed animal, a stressed gut, that this axis between the central stress response is acting on mast cells, and we think that this is a critical part in regulating permeability. Here you can see CRF activation in the gut elevates permeability, and it also elevates pro-inflammatory cytokines, and that this is mediated through a mast cell dependent mechanism. So to begin to tidy this up here, we've shown that when there's an early life stress or early life gut injury, there are stress response systems that are activated in the gut, causing immune cells, such as the mast cell, to become activated. And we think that this is really integral to the long-term gut barrier function and development of the gut, and heightened stress responsiveness, and also is dictating how that animal will respond to a stress or an infectious challenge later in life. So to end, to state again, there's been a lot of work on pathogens and susceptible hosts, which is still really relevant in disease because the more we understand the mechanisms on how pathogens influence the host, there's more opportunities for therapeutics in there. But the one area that my program has been focused on, we're finding a lot about, is how this other side, this lesser studied area, which is represented by a smaller arrow, is how when pigs are in these environments, how does that shape the animal and how they're going to respond, not only at an early point of their life, but potentially through how they're going to be programmed to respond for the rest of their production span.